So I'm going to talk today about whether satiety has a role in energy balance. So to start off with, I'm from the Functional Food Centre at Oxford Brookes University. This is our commercial research centre. So what we do is we work with food companies. If I speak too fast, please let me know. I talk quite fast. I'll try to slow down. So we work with food companies and we try to meet the needs of food companies in terms of the nutritional research that they want to do. Our vision is to work with companies to improve the health and well-being of the global population. So we have three main key areas that we work within. The first one is metabolic testing, where we focus on glycemic response and control, looking at energy expenditure and diet-induced thermogenesis. And we also look at things like gastric emptying, gut hormones, and lipemic responses. Our second area is weight management. So here we're looking at weight loss studies and things like that, body composition, energy regulation, and appetite control. And our third area is looking at food components and activity. Things like bioavailability, bioaccessibility, antioxidant status, and things like that. So here you can see how we work. We have our three main areas here. Our aim being to increase health and well-being. And we work in novel food ingredients, novel foods, and putting these all together into whole diets. So, if anyone would like to find out more information about the Functional Food Centre, I've just left some brochures here. And we're also having a conference in November, so I left the brochure for that too, if anyone's interested. So let's put all this into a bit of context. Firstly, we're going to look at why we might want to study satiety. Then we're going to examine what satiety actually is, how it's controlled, and how we might go about measuring it. And then later we're going to explore, later on this afternoon, we'll explore some functional foods, primarily functional lipids. So, this is data taken from the UK. It's taken from the Health Survey for England, and we can see the statistics for obesity levels within the UK. So in men, you see the healthy people are only about 32%, whereas obese is 24%, and the majority of the population is overweight. Similar statistics in females, slightly better, more healthy females and less overweight. And this isn't something that's just happened overnight. This thing has been progressing for many years, and we can see here in America, data taken from the US National Health and Nutrition Examination Survey shows the increases in obesity levels between 1976 and 1980 up to 1999 to 2000. And we can see across different ethnic groups, we can see the increases in obesity, both in men and in women. And because of this growing trend of obesity, the Centre for Disease Control in the US has predicted that by 2059, every obese person will be obese if things continue the way they are, which is quite shocking, I think. And just to bring this back home, because I know not everybody lives in the UK or lives in America, this is data taken from Europe. And it shows the growing trends. Some of you have probably seen these slides already across Europe. So in 1985 to 1989, there wasn't much data from many countries. The UK is included on here, with a low percentage of obese people, 5 to 10%. As we go up to 1990 to 1994, there's more data for a lot more countries. You can see Spain here in 10 to 15%, and females slightly higher. Actually, what happens next for females in Spain is actually quite interesting. So if we go on further, we can see the majority of countries are getting a lot darker. France, Ireland, Italy, and Greece down here, all becoming into the higher percentages of obesity. What's interesting is that if we go back to here, you can see that in the earlier years for females in Spain, it actually is higher, and then as we go further on in years, it actually gets less, which just kind of bucks the trend a little. But generally across Europe, we see that it just gradually gets darker and darker, and the numbers are getting much worse. To the extent that by 2000, 2008, we see that some of the countries here are in the over 25%. So we see Greece here, I think Scotland over here, I think Croatia there, and Albania down here. So a lot of these countries are now in the over 25% of the population are obese. So there's one bit of good news. In 2010, a study by Flegel et al. Showed that the BMI is beginning to plateau. So if you see here, you can see that the levels actually aren't increasing very much. They're just starting to plateau, both in men and in women. So this is obviously good news, but in saying that, the IASO and the International Obesity Task Force still estimates that 1 billion people in the world are currently overweight and 475 million are obese. So there's still a huge problem that needs to be dealt with. So what's the impact of this? So, that's fine, people are overweight. Does that actually mean anything? Like, okay, maybe they're just not as pretty. Fine, if that's what people think. But, what does that, this has other implications. Aesthetically, it doesn't really matter. But, how does this impact on their health? So we can see that at different body mass indexes, we see that the rate of mortality at low body mass indexes is higher, but as we increase our body mass index, the rate of mortality increases. And the relative risk of developing diabetes at higher body mass indexes starts to increase also. Similar with other diseases, we can see here the relative risk of being overweight and obese in men and in women. So the relative risk of all-cause mortality increases as we get obese. The relative risk of ischemic heart disease doubles with obesity. And if we go down to diabetes, we can see that it goes up to 5.5 times or 7 times the risk. So, energy balance. You should all be able to tell me about energy balance, I'm sure, at this stage. Obviously, with energy balance, we have energy in and energy out, and we want the two of them to match up so that a person stays in energy balance. Energy in comes from our food, from carbohydrates, lipids, proteins, and alcohol. And energy out comes from 3 different sources. So our basal metabolism. Can anyone tell me what basal metabolism is? No? Yeah, so how much energy you need to sustain your basic metabolic functions. Then we have thermogenesis, or diet-induced thermogenesis. Anyone want to tell me what that is? Exactly, you should know. So it's the amount of energy that's needed to digest, absorb, and assimilate your food. And then obviously physical activity. So physical activity can be anything from just sitting there, writing notes, or whether you go out for a run, but anything above what you need for your basic bodily functions. So what's interesting is that energy expenditure is through several different methods, but there's only one mode of food intake, so that's through food and drink. So the thing is that energy expenditure is continuous. You can't stop yourself expending energy, but you can stop yourself eating food. So where energy expenditure happens all the time, food intake doesn't, or energy in doesn't. Food intake is self-controlled, and absence promotes search and ingestion. And the quantity of food is obviously self-limited, which is what makes it quite interesting. So food has nutrition, social, and cultural roles. However, its principal function is to give the energy needs to the body. So what are our primary reasons for food choices? Why do we go about eating? It's not just because we become hungry and we want to eat. There's a whole load of other inputs that need to be taken into account when we talk about food choice and consuming food. Things like advertising. So targeting children, you see advertisements that primarily target children. So when they go to the supermarket with their parents, they can go, oh, Mum, I want that. Availability. So if you're a student and you have only a certain food in the fridge, you might just have that food because it's the only food available. Economy. Again, if you're a student, this might be a priority. Emotional comfort. For example, women like chocolate a lot of the time, maybe, for emotional comfort. Habit, perhaps. It's something that you always eat first thing in the morning, something you always do at a certain time of the day. People obviously have preferences for foods that they find palatable, so they will consume them. Positive associations, being at the beach. People like to eat ice cream when they go to the beach, so they have a positive association with ice cream. Regional preferences. So For example, I'm trying to think of an Irish regional preference. I'm actually Irish, so I don't know, maybe bacon and cabbage or something like that. But we have a wide variety of people here from different countries, so I think I should get a few different examples of regional preferences. Can someone tell me a Spanish regional preference? Paella? Yeah. How do you pronounce it? Paella? Paella. Okay, I'm not going to try it. Sorry? Potato omelette, yeah. Tortilla? Is it? Yeah. Any other countries we have here? We have, I believe, we have people from Poland. Yeah? What's the regional food in Poland? Pierogi. Do you want to explain what it is? Alcohol, okay. Same in Ireland, so. Is that what you said? Okay.
Pierogi is okay when you put tomato, not tomato, but potatoes with white cheese.
Okay, inside.
Onion, yes, salt,
Oh, sounds nice. Dumplings, okay. And any other countries here? Yeah? Guatemala. What's your regional preference?
We like beans, avocados, and corn.
Oh, nice. Tortilla, nice. Any other countries? Mexico? What do you like? Chile? Tortilla, yeah. Yeah. Cool. Okay, so let's move on to the next one. They're good examples, though, of, if you're in a country as well, you might want to try out these regional preferences, and that might be your reason for food choice. Other things like social pressure to try out certain types of food, your values or beliefs. So traditionally in Ireland, people didn't eat meat on a Friday, which is a Catholic thing to do. Perhaps some people might have just finished Ramadan. Things like this, or also people's beliefs about, not consuming animals and being vegetarian, things like that. Food choices associated with weight and health. So perhaps if you are allergic to certain types of food, if you have your doctors put you on a certain type of diet, you'd make food choices based on this. And also to do with the nutritional value of a food. So if you believe that a certain food is high in something, then you might choose to eat that. So the main factors that determine the size and rate of eating a meal are hunger, appetite, satiety, and satiation. Hunger is the feeling that occurs when an individual wants to ingest food. So it's a feeling of food deprivation that signals when the next eating episode should happen. And it's distinguished from appetite, where appetite is a psychological desire to eat. So it's associated with orosensory acceptability of food. So where hunger is more of a physiological response because you feel hungry and you want to eat something, appetite is a psychological response. So for example, I could be walking around here and there's some fantastic bakeries I've noticed, and as you walk by some of them, you might see the produce there, and I've just had lunch but I'm really, really full, but I still want to eat them. That would be appetite. Whereas hunger might be, I could be hungry without having appetite because perhaps I've been sick and I don't actually feel have any appetite to eat, but I do feel hungry. So that would be hunger. And in order to have food intake at an adequate level to meet your physiological needs, both these things need to be present. So you need to be hungry and you need to have an appetite, which is what most of us are most of the time. Well, I hope we're not hungry all the time, but you know what I mean. So the frequency and length of a meal are governed by satiety and satiation. The termination of a meal as a result of inhibitions of feelings of hunger and appetite is what satiation is. So it's when we stop eating a meal. So during a meal, you're eating it and you have a massive plate, big plate of food, and you stop eating halfway through it because you have satiation. Whereas satiety is the inhibition of feelings of hunger and appetite in the post-meal phase. So when you stop eating, it's what stops you eating another meal. So while satiation will determine how much you eat in a meal, satiety will determine when the next meal is consumed. That makes sense? Yeah. Okay, well, stop me if you have any questions. And satiety and satiation are described in what's known as the satiety cascade, which was developed by Blundell in 1994. So here we can see satiation within the meal and satiety in the post-meal phase. Satiation and the early stages of satiety consist of the sensory and cognitive phases. Following this, we have a post-ingestive phase and then a post-absorptive phase. And here we can see it described in more detail. So we can see the sensory and cognitive associations refer to the meal quality, our expectations about the meal, what we feel, how we feel it should taste, it should feel, and our prior associations and beliefs about it. Then after this, we have the post-ingestive phase, which occurs in the stomach and intestines and is associated with meal quantity. So things like the release of hormones and distension of the stomach. And following this, we have the post-absorptive phase, where the nutrients are actually absorbed. So the sensory and cognitive phase talks about your previous experience. So if you consume a certain meal, and you know what that meal tastes like, you have an expectation about how it will taste again. So if you go to pour yourself a bowl of muesli in the morning for breakfast, the next day you will have, and you know that amount will fill you, the next day you will go and make a bowl exactly the same size because you know that's the amount that fills you, and you have an expectation that that is what will fill you. The same if you go to a bakery, you have an expectation that the food, the smell that you get from the bakery will invoke the same feelings of taste and palatability as the smell does. So you have an expectation in terms of the reward and pleasure associated with the food and the palatability of the food. There's another thing that happens within the sensory and cognitive phase, a physiological response, and this is known as the cephalic phase response. So this response occurs in anticipation of food. So before you actually consume a food, this response is actually happening. So before you actually consume a food, you have an expectation, as we said, about it. So if it's palatable food, it might cause you to increase salivation in your mouth. It can cause the release of insulin, and it can cause gastric acids to be released. All of this happens before you've even put the food into your mouth. So you're getting an insulin response even before any glucose has been taken in. And here you can see, down here we see time, and here this arrow indicates when the meal was starting to be consumed. And minutes after the meal is consumed, we see this peak in insulin. And this is where the actual insulin increases with the glucose. So this peak here is a cephalic phase response to the ingestion of the meal. Or just having the meal in your mouth. And similarly here, we see this is when the meal was started to be consumed, and we see this rapid increase even happening before the meal was consumed. So how does the cephalic phase respond to satiety? What impact does it have? So the effect of cephalic phase responses to food intake have been debated, because on the one hand, they increase saliva secretion and they increase gastric accommodation, and they get the body ready to consume the food. So in essence, this should increase food intake. But they also cause an increase in insulin and another hormone called pancreatic polypeptide. And these two hormones increase satiety, so they should reduce food intake. So here we see pancreatic polypeptide levels in response to what's called sham feeding. Sham feeding is where you eat a piece of food, put a piece of food into your mouth, chew it, and then spit it out. And then you do that continuously for an entire meal, so you don't swallow any of the food at all. And here you see this increase in the satiety hormone, even though no food is being eaten at all. So whether cephalic phase responses increase or decrease satiety is unknown. Okay, so we just talked about the sensory and cognitive phases, and we're going to move on to the post-ingestive phases. Post-ingestive phases consist of gastric distension, gastric emptying, the holding time in the stomach, and the osmotic load. They also have food factors, afferent signals, and also hormones associated with the stomach and small intestine. I'm going to talk through each one of these individually, so I'm not going to go into the detail now. So we're going to talk first about the first one, gastric distension, gastric emptying, and holding time. So when we get food into our stomach expands. This causes gastric distension or expansion of the stomach, and this signals fullness via the vagus nerve and tells our body that, the stomach is full and it's time to stop eating. This gastric distension, the stomach itself can't sense what type of nutrients are in it, so it doesn't know whether the food in it is fat, carbohydrate, protein. It doesn't know any of this. It just knows the volume of food that's in it. However, when we have gastric distension and food being delivered into the small intestine, this increases the feelings of fullness or satiety even further. So here we see an example where we have a fullness score given following different volumes put into the stomach. So what they do in this case is they put a tube down into the stomach. At the end of the tube they'll have a balloon, and they'll fill the balloon with different volumes of water. So here we see the different volumes that they put in. And we can see with no volume there is no change. Then as it increased to 400 millilitres it increases, and then at 600 and 800 millilitres it increases even further. And then what they did after this was they infused the duodenum with nutrients, and we see that the fullness level goes up even further. So we can see here that the stomach does play a large role in giving signals of fullness to the body. Another example of a food where we can get gastric distension. So we all know, probably, I hope anyway, about that water can increase the volume of food. We can use fibre to increase the volume of food, but we can also use air to increase the volume of food. So in this study what they did was they made different milkshakes, and in the milkshakes were exactly the same, but they incorporated air into the milkshakes, so that one milkshake was 300 millilitres, one was 450 millilitres, and one was 600 millilitres. And then they had one where they just fed an ad libitum meal. They didn't have any milkshake. And we can see with the increase in the volume of the milkshake, there was a decrease in the food intake. So just by expanding the volume of the food, they didn't even change the nutrient density of it, because nutrient density is obviously per gram of food, and the weight would be the same because air doesn't weigh anything. So all they did was incorporate air, and they found that the energy was decreased. So other post-ingestive factors. These are food factors, things like viscosity and particle size, and the liquid-solid state. I thought a really good example of post-ingestive satiety is soup, because soup kind of contradicts everything that we know about post-ingestive satiety. So we know that solids are much more filling than liquids. So if you think of fizzy drinks, we know that fizzy drinks are really bad because people take on a lot of nutrients in a liquid which isn't very satiating or very filling. But soups are liquid, but they are really filling. So it's kind of a contradiction to what we usually believe. And there's been a lot of studies carried out looking at soups and how they affect satiation, but not many have actually studied why they affect satiation. So here's one study showing an example of soups decreasing food intake, and we can see they had a control, a casserole with water, and then a soup, and soup decreased food intake. So when we look at gastric emptying, or emptying of food from the stomach, what happens is we see here the percent of the meal retained in the stomach, and we see for solid food and for liquid food. For solid foods, there's this what's called a lag phase, or a delay before the food actually starts emptying from the stomach. And this lag phase is whereby the food is broken down into particles that can be emptied from the stomach. And then after that, it decreases in this linear way. Whereas for liquids, it empties much more rapidly in this exponential fashion. So that's why liquids tend to be less satiating, because they empty so fast from the stomach. However, we compared soup, and what we did was we looked at a smooth soup where we blended everything. We looked at a chunky soup where we had particles inside the soup, and then we looked at a solid meal. And they're all exactly the same meal, but they were just blended up to varying degrees. And what we found was that the gastric emptying times were much longer for the soup compared to the solid meals or the chunky soup. So here you can see time for half the meal to empty from the stomach was 97 minutes for the smooth soup, whereas for the solid meal it was only 86 minutes. So the soup itself, because it becomes so viscous and thick, it actually empties slower than a solid meal itself would. So next thing we're going to talk about is afferent signals, so associated with food quantity and nutrient composition. So we talked about food quantity a bit, talking about gastric distension and fullness. So I'm going to focus on nutrient composition. And the effect of different macronutrients on satiety and satiation has been well studied. And this is just an example of one study that I'm just going to talk about. This study by Johnson and Vickers, they fed 3 different meals. One was a pasta meal, which is primarily carbohydrate. The second was a whipped dessert meal, which is primarily fat. And the third one was a chicken meal, which was primarily protein. And they fed them in equal energy content, and then they examined their food intake at a buffet meal afterwards. And this is the results from the buffet meal. So what they found was that eating the high protein or high carbohydrate preload decreased hunger compared to the fat. And then eating a high protein preload decreased the mass of food eaten more than the high carbohydrate or high fat preload. So if we look down here, we see the pasta, whipped dessert, and the chicken. They actually ate 2 different meals for each one. So one of them was 150 kilocalories and one was 300 kilocalories. But we'll focus on the 300 kilocalorie one, this one, and this one. And we can see that the amount eaten was 357 for the pasta carbohydrate meal, 409 for the fat meal, and even lower, 345 for the protein meal. So this shows us that there tends to be this hierarchy in terms of macronutrient satiety. Now, can anyone tell me why there might be problems with some of these studies? Anyone know? What's different between protein, fat, and carbohydrate? I heard some whispers. Energy density? Yeah. So they all have a different energy density, or fat has a different energy density. How many kilocalories, someone on this side, is there in fat? 9, yeah. And protein? 4. And carbohydrate? Yeah. Okay, so if you look back to this study here that I just showed you, we can see that the energy content is the same, but the weight of the meal isn't the same. So obviously there's going to be an effect on this gastric distension we talked about earlier, because the volume of meal that they've consumed isn't the same. Now, there has been some studies that have controlled for this by giving things like soups, where they can just add extra water in to control the volume. And the results haven't been as clear in terms of macronutrient satiation. There have been some that have shown the same hierarchy, but some that haven't. However, there are problems associated with eating, obviously, a high-fat diet and this macronutrient satiation issue. So Liz et al. Placed people on a 14-day diet that was ad libitum. They're either 15 to 20% fat, 30 to 35%, or 45 to 50%. And they could consume as much as they wanted of the foods that were available. What they found was the energy intake increased depending on which diet. So it increased from 2,090 up to 2,350 and up to 2,700 from the low-fat to the high-fat diet. And this was associated with weight changes as well, that the low-fat diet lost weight, whereas the high-fat diet increased their body weight. And this is primarily due to the fact that the energy density of fat is much greater and people don't compensate as well for the amount of fat that's in food. So the last factor, I think, on post-ingestive satiety is associated with hormones in the small intestine. The hormones that are secreted in the small intestine are secreted in response to nervous activity, so stimulation from the nervous system, such as by the vagal nerve that we talked about, distension of the stomach, and by chemical stimulation. And this chemical stimulation could just be the nutrients that are present in the small intestine. They're concurrent with food intake, so they change in response to food intake. And the gastrointestinal tract produces more hormones than any other organ in the body. So the first one we're going to talk about is cholecytokinin, CCK. And CCK is secreted from the mucosal endocrine cells of the small intestine. It's secreted primarily after fat and primarily after long-chain fatty acids, also after protein, but not so much after carbohydrate. It has a powerful influence on gastric emptying, so it delays gastric emptying, which can obviously cause increased feelings of fullness in the stomach because the food gets delayed there for longer. It also increases pancreatic secretion and increases bile secretion. So if you think about it, it's secreted in response to fat. We know that we need certain things to digest fat. What do we need to digest fat? What do we need to digest fat? What do you say? Bile? No. Enzymes? Pancreatic enzymes, yeah. So, in other words, we need bile to emulsify it, and we need the pancreatic enzymes to digest it. So, when the fat comes in contact with it, it increases the release of CCK, which increases the release of this pancreatic secretion and bile secretion, which all help in the digestion of fat. So it's this chain reaction in order to digest the fat. It also promotes satiety and decreases food intake. Because if the fat is coming into the body or the food is coming into the body, it's telling the body basically that it doesn't need to eat as much food. So here we see the response, CCK response to different nutrients. And we can see in response to fat, there's this large increase. In response to amino acids, there's also quite a large increase. But if you look at the scale here, you'll see it goes from 0 to 6, whereas for fat, it goes from 0 to 8. And we see the response is lower, and even lower again for glucose. And what we see also here is this plasma CCK secretion in response to a meal. And we see after the meal, there's a large peak in CCK levels, and this also corresponds to a decrease in gallbladder volume. So in other words, the secretion of the bile. So as the CCK levels start to increase, the bile starts to be secreted from the gallbladder. The next hormone I'm going to talk about is ghrelin. Ghrelin is produced by the exlymptic glands in the stomach and also in small amounts in the small intestine, but primarily in the stomach. Ghrelin is quite novel in that it's secreted in anticipation of a meal and it increases food intake. So it's the only gut hormone that increases food intake. All the rest of them decrease food intake. And it's believed to have a role in the timing mechanism. So it will tell the body when it needs to eat next. And it forms a complementary balance to leptin, in that leptin does the opposite effect. Ghrelin plays a role in gastric motility. So whereby CCK delayed gastric emptying, ghrelin will increase gastric emptying and get the food to move more rapidly from the stomach into the small intestine. So because it increases the hunger levels, this makes sense because you eat the food and you want it to get rapidly into the body, so it increases gastric motility to do that. It stimulates food intake and decreases energy expenditure. And here we see, this is quite a famous study by Cummings et al., and here we see its role in timing mechanism. So we see the ghrelin levels start to increase, and this dotted line here is a meal, and then it decreases following the meal. Increases again prior to the next meal, and then decreases after the meal, and the same after the next meal. So in other words, it increases prior to meals so that you take food on board, and then decreases after that. Here we see its effects on gastric emptying. We see gastric retention, so the amount of food that's retained in the stomach. And we see that after saline, there's this emptying like this, whereas after ghrelin, it's much more rapid. So the food empties from the stomach much more rapidly. And here we see the hunger ratings, and we see that the ghrelin increases the hunger rating, whereas the saline doesn't. And similar for the desire to eat rating. Next hormone is glucagon-like peptide, or GLP-1. GLP-1 is primarily secreted in the small intestine, but also in the brain. It's secreted in response to meal ingestion and primarily to lipids and carbohydrates. And what it does is it adjusts the small intestinal digestion in order to meet the ability of the stomach to digest and absorb the food. So this is known as the ileal brake mechanism. And the ileal brake mechanism is basically a brake, as in it slows things down. So it's a feedback mechanism from the ileum that causes everything further up the gastrointestinal tract to slow down. So it delays gastric emptying, it halts the secretion of bile and pancreatic secretions, and it increases satiety to stop the food being taken on board. And here we see just some data from it as well. This data shows the percent remaining in the stomach, and we can see that following GLP-1 secretion, we see that the amount remaining in the stomach is much greater compared to a saline infusion where the meal empties quite linearly here. And similarly, we can see with increasing amounts of GLP-1 from control up to 1.5, we can see that the food quantity is reduced from 587 up to just 384 following the GLP-1. And this study by Vergist shows that GLP-1 infusion reduces energy intake in both lean and in obese individuals. So that obese individuals are still susceptible to the effects of GLP-1. So we can see that the higher the rate of GLP-1 secretion, that the lower the amount of energy that's taken in. So at the lower levels of higher levels of GLP-1, there's less food intake taken in. At the lower intakes, there's a greater amount of food taken on board. And one of the reasons that this is thought to occur is because, as we talked about before, we have this brake mechanism which really strongly stops movement through the small intestine and the stomach, which increases the amount of food remaining in the stomach, increases this gastric distension, and causes this feeling of fullness. The final hormone I'm going to talk about is PYY, peptide tyrosine, and it is secreted from the L cells in the ileum and colon, so the distal end of the small intestine. It's secreted 15 minutes after ingestion, and it's released in direct contact with nutrients. So one of the reasons that these two hormones, this hormone also plays a role in the ileal brake mechanism, and one of the reasons that these two hormones work is because they both are done in response to food in the ileum. So if there's food in the ileum, we know that the ileum is the last part of the small intestine. So there shouldn't really be any nutrients left there because everything should be absorbed at that stage. So it's a feedback mechanism to basically break everything so that the food, that everything can control further up to get time to digest and absorb the food. And so it has exactly similar responses to GLP-1, delays gastrointestinal transit, reduces food intake, and increases satiety. And here we just see a couple of examples. We can see changes in hunger scores, and we can see with saline that the hunger scores increase, whereas with PYY the hunger scores stay low. They don't become hungry. And here we see the change in food intake. So with saline, there's a large intake of food, whereas when PYY is infused, there's less food is taken on board. Okay, so I'm just going to recap slightly on the four hormones I talked about. First one was CCK. Can anyone remember what happened with CCK? What does it do to food intake? Increase or decrease? Decreases food intake. What does it do to gastric emptying? Increase or decrease? Decreases, yeah. And what other things were there? Let's find out. Oh, bile and pancreatic secretion. So it increases gastric emptying, increases food intake. The only orexigenic gut hormone. And GLP-1, it plays a role in the ileal brake mechanism, so it causes a brake on everything and stops everything. So it stops pancreatic secretions, stops gastric emptying, stops bile secretion, and reduces food intake. And finally, PYY decreases food intake, decreases transit through the small intestine and stomach, and decreases bile secretion as well. So, we've talked about the sensory factors and cognitive factors, talked about the post-ingestive factors. We're just briefly going to talk about the post-absorptive ones. These are associated with signals arising from when the food has been digested and when it's been absorbed in the small intestine. and the signals that are released from this. So things like the release of insulin in response to glucose, increases in amino acids in the blood, and all of these will cause satiating factors, which will have a longer term satiation. So they occur after things have been absorbed rather than when things are still within the digestive tract. So how do we go about measuring hunger and satiety and food intake? Well, studies can either be free living or they can be laboratory based. And there's advantages and disadvantages to both. In free living studies, we have less precision and accuracy and less control, because if you bring volunteers in and then ask them to go off and do their normal activities, you can't control that they tell you everything they're going to do, that they do everything that you want them to do, and you don't have as much control over what they're actually doing. You can't keep an eye on them all the time, as much as you could try. You can't. So, but there's good advantages to it, because they're going about their daily lives, so they're doing normal things that normal people do. People don't spend their lives in laboratories, but they do spend their lives, going to the beach or going to classes or going to work. And you can get, from this, you can get what people actually do in their normal daily lives. So from this, you might be able to see, if they have a snack at a certain time, whereas if you came to the lab and you tell them when they have to eat certain things, then you're not going to get that. And also you don't get things like we talked about earlier, the different things that cause people to make food choices, all like advertising that's attacking people all the time. They don't get that in a laboratory situation. Whereas when they're outside living, all these factors are taken into account. In a laboratory, obviously you control when someone's going to do something. You can tell them when you want to fill in a questionnaire, and you know that they've done it at that time. You can tell them when you want them to eat something, you know they've done that at the right time. So you have a lot more control over what they do. But again, it doesn't represent what they might do in their daily life. So there's different ways in which we can measure satiety and hunger. These are visual analog scales, actually measuring their food intake, so through a preload paradigm, or through self-reported food diaries, or through biochemical measures. So I'm not going to talk too much about the biochemical measures because we've kind of touched on them already. I'm going to focus on the top two. So visual analog scales, these are subjective measures where you ask someone to rate how they're feeling in response to something. So they're used a lot in satiety data, but they're also used in other aspects like pain and measurement and things like that. There's two standard methods that are used. There's a continuous line visual analog scale here, which are usually 100 or 150 millimeters in length. Or there's hedonic scales or category scales. And these are broken down into different categories that a person can rate how they're feeling. The continuous lines are generally considered better because it gives the person the chance to use the entire length of the line to make their decision on. However, you have to remember that these are subjective. So what one person says can only be compared to that person. So if I measure you, I can only compare that data from you to you, because he might use the line in a different way to how you use the line. So, for example, one person might always come in and be like, I'm starving, and they tick right down the very end. Another person might come in and say, oh, I'm starving, but they tick 10 millimeters up the line. It's just how different people react to different things to it. So it's completely subjective, and you can only compare from within one person. The next type of measurement is, this shows how you use them. So the general questions that are used for visual analog scales are: how hungry do you feel? How full do you feel? What is your desire to eat? And how much food do you think you can eat? And these questionnaires are pretty standardized across the world. So people generally will use these 4 questions. Or maybe a couple of variations of them, but these questions are what's standardized and used, which is good because it means that you can have comparisons between different studies then. The preload paradigm is whereby people have given a preload of a food or maybe a sample of a food. Then a certain time passes and they're given ad libitum intake or free choice of a range of foods to eat as much as they feel they can until they feel comfortably full. And this gives an actual measure of food intake. So whereas visual analog scales are just their interpretation of how hungry they feel, this is actually a measure of how hungry they actually feel. One thing about using the preload paradigm is that you can't standardize it. So we just talked earlier about regional variances in food. And what you have in Poland as a normal buffet meal, you couldn't have in England or you couldn't have in Mexico, because people in different countries eat different foods, and you can't have the same comparisons between meals. Also, it can be difficult to choose the different types of foods you're going to do, because you think that people will really like a certain type of food, but they might not actually like it. So, for example, I thought that when I did this study, that Irish males all like to drink milk. No one drank milk. Not one subject drank any milk during the whole study. And every day I measured out milk, but no one drank any of it. So you ask people at the start to rate how much they like each food, and you only allow them into the study if they say rate, over 70% of the foods as liking them. Everyone liked milk, they just didn't want to eat milk or drink milk. So here is how a typical study would happen. People would come in after an overnight fast. They'd have a visual analog scale, and then they'd consume a breakfast meal. This breakfast meal would be standardized, so they'd eat the same one each time that they came in. They then would fill in another visual analog scale. After a certain time period, they would fill in another visual analog scale and then consume the test food or preload. Now, depending on what time within the satiety cascade you want to measure, you would then give them a lunch after this. So if you wanted to measure the sensory and cognitive aspects, you might perhaps have a preload and then have a dessert. So it would give you a meal, and then dessert would be ad libitum, so they could eat as much as they liked. And this would give you the sensory and cognitive aspects. Whereas if perhaps you wanted to look at the post-ingestive phases, you might look at it 30 or 60 minutes afterwards. Whereas if you wanted to look at it the post-absorptive, you would do it later than that. So depending on which time period within the satiety cascade will depend on the time period here. So self-reported food diaries. Self-reported food diaries are often used both within the preload paradigm and also within prospective studies. So for example, if you have someone come into the lab, do a preload paradigm, as we did before, just here. Then after they have their ad libitum lunch, you might ask them to leave the laboratory and take their food diary with them and fill it in for the rest of the day. Or food diaries obviously can be done at other times. So if you want to get an indication of someone's food intake, you could ask them to use a food diary for 1 weekend day and 2 weekdays. Obviously, the greater the amount of time you have on a food diary, the more likely you're to get an overall picture of their entire diet. You obviously ask them to give them instructions on how to write it and try to make sure that they give as much detail as possible. So you can see here they have the food and drink, a description of the preparation of the food, and a description of the food. So it's very easy for people to write down milk instead of saying semi-skimmed milk or full-fat milk. They could say that they ate carrots, but they don't say what way they cooked the carrots or anything like that. And so it's really important that they give as much detail as possible. So here's the advantages and disadvantages of each type of method. For visual analog scales, they're easy and quick, so it only takes 2 seconds to tick a line on 4 different questions. They're cheap because all you need to do is print out some paper. There are handheld portable monitors that can be used nowadays, but you don't have to use them. You can just use a piece of paper. They're simple to interpret because they're 100 millimeters in length. All you have to do is get a ruler, measure the length across it, and that's it interpreted. They have a standard format, the 4 questions we talked about, and they're data on subjective feelings, so they give an indication of how that person feels. However, they have limitations. The accuracy of the data, because it's subjective, it's only an indication of how they think they feel. It's not an indication of how much they would eat. It has a low sensitivity, And it's a non-blind method because they know you're asking about how much they're eating. You know, they know that you're want to know about their hunger levels. Whereas, say, for the preload paradigm, you can tell them, oh, have as much food as you want to eat, but you've got to stay in this room for a certain amount of time. So if you do that, they have to stay for a certain amount of time, and you'll get an indication without actually telling them that you're measuring their food intake. The preload paradigm is good because it's reliable data. It shows you how much they would actually eat. It reflects their actual physiological needs, and it can be a blind method. However, it's costly and time-consuming. It obviously takes a lot of time to measure out all the different types of food, weigh them beforehand and weigh them afterwards. And providing that amount of food every single time tests, and you need to make sure that you provide it all in excess so that they can't run out of any type of food. You need to have correct preferences of the food available, and you need to have enough portion size and amount offered. It cannot be standardized between countries, and the food intake itself can be influenced by a variety of factors. So if you have somebody in the laboratory and they get distracted by somebody else there, they might eat less. If you have, for example, if the room is too hot, they might get affected by that. If the room is too cold, they might get affected by that. So a lot of different things that you wouldn't think would impact on your study could quite easily do that. Self-reported food diaries are convenient because they just ask somebody else to do a lot of the work. You don't have to do it. So they allow you to just pass that responsibility on to them. And they're cheap, obviously, because all you need to give them is a weighing scales and the food diary. However, they depend on the person to give an accurate and complete indication of their diet. And this, we know, doesn't always happen. People tend to leave out bad things if they know that they've eaten bad things. And this can obviously give a wrong set of data to you. And they can be difficult to analyse. Inputting food intake data can be quite tedious and take a lot of time. So, what did we cover today? We defined the issue and we looked at the obesity levels across the world. We've shown that they're obviously quite high and that we have an issue here. And that this issue is associated with a long list of different diseases that can influence our mortality levels. We defined appetite and hunger, and we looked at satiety and satiation. And we defined these. Can anyone tell me what appetite is? A desire to eat specific food. Yeah, so it's more of a psychological desire to eat a food. And hunger? Sorry? Yeah, the physiological need to eat. Exactly. Good stuff. And then we looked at the different aspects of the satiety cascade. So the sensory and cognitive aspects associated with our preconceived ideas of how a food should taste, palatability, things like that. And we looked at the cephalic phase responses. We then looked at the post-ingestive phases, so things to do with food factors, particle size, gastric distension, gut hormones, and macronutrients. And then we looked at different methods of measuring satiety.